For a healthy human or typical mammal, the baseline fasting blood glucose level sits tightly regulated between 0.7 and 1.0 grams per liter. This is the biological standard for safe metabolic function. But if you look at a healthy fasting bird, like a chicken, that baseline is naturally between 2.0 and 2.15 grams per liter. If a human sustained blood sugar levels that high, it would trigger severe, fatal diabetic organ damage and rapid systemic failure. Yet not only are birds perfectly fine at these levels, it takes massive, near-lethal doses of injected insulin just to force their blood sugar down into a state of basic hypoglycemia. Chickens operate at these levels with zero evidence of organ damage, proving that whole-body insulin resistance can exist as a stable, healthy state rather than a sign of a broken system. In medicine, massive systemic insulin resistance usually points to a clear structural culprit. Either the insulin molecule itself is defective, or the cellular receptor it docks into has mutated. The avian insulin molecule is completely intact. Its sequence is highly conserved across species, except for one specific substitution. On the A chain, exactly at position 8, avian insulin trades the mammalian threonine for a single histidine residue. That tiny chemical tweak doesn't weaken the hormone. It turns chicken insulin into a natural superanalog. Avian insulin binds with significantly higher affinity and a much slower dissociation rate than human insulin. It grabs the receptor tighter and holds on longer. And the avian insulin receptor itself functions perfectly normally the moment that bond occurs. So the ligand is hyperpotent, and the receptor lock works perfectly. The systemic resistance isn't happening at the surface. It is originating from a deliberately rewired circuit deep inside the cell's cytoplasm. Looking at this diagram of the cellular signaling network, we can trace the insulin signal past the outer membrane and into the cytoplasm. In mammals, the primary signal transducer directly beneath the receptor is insulin receptor substrate 1, or IRS1. In the avian liver, this pathway works exactly as expected. IRS1 phosphorylates and transmits the signal normally. But in avian skeletal muscle and adipose tissue, IRS1 is profoundly unresponsive to the receptor. Whether the bird is fully fed or fasting, the tyrosine phosphorylation of IRS1 in those specific tissues remains totally dormant. The initial signal translation through IRS1 is completely blocked in avian muscle and fat. This is the first major point of systemic resistance. To find the source of that dampening effect, we have to look further down the cascade from IRS1. Here, further down the pathway diagram, we find PI3K. This is the critical metabolic gateway required for glucose uptake and lipid regulation. Here, we find a startling anomaly. Basal PI3K activity in avian leg muscle is roughly 30 times greater than the baseline activity found in rats. Because of this constitutive hyperactivity, the kinase is running at maximum capacity long before any insulin even arrives at the cell surface. That chronic overdrive triggers a relentless feedback inhibition loop, which actively desensitizes the muscle to new inputs. The avian tissue actively deafens itself to the super-analog insulin. It ignores the signal because its internal PI3K machinery is already redlining in a state of permanent overdrive. This creates an obvious physiological problem. If the primary IRS1 pathway is blocked and the PI3K gateway is deafened, how does insulin actually manage to keep the bird alive? The avian system circumvents the blockade by employing a highly specific evolutionary workaround, recruiting an entirely different protein to carry the necessary survival signals. Looking back at the receptor diagram, we see the SHC protein attached directly to the beta subunit. Birds utilize the 52 kilodalton isoform of SHC to completely bypass the broken IRS1 pathway and push the signal forward. In the avian liver, this specific SHC isoform acts as the primary dynamic responder. Its activity directly fluctuates based on whether the bird is in a feeding or fasting state. This stands in stark contrast to mammalian systems, where IRS1 universally dominates that metabolic role. In fact, this unique direct association between the 52 kilodalton Shek protein and the PI3K regulatory subunit is essentially uncharacterized in mammals. This Scheck-mediated bypass represents a precisely engineered adaptation, allowing the bird to maintain its necessary survival signals in a state of constant metabolic extremity. To understand why this internal extreme exists, we need to pull back from the microscopic signaling pathways and look at the macro architecture of the bird's organs. This anatomical diagram illustrates the unique physical structure of the avian pancreas, stretching across 4 distinct lobes alongside the duodenum. If we look deep inside that endocrine tissue, into the islets of Langerhans, a striking cellular makeup emerges. We find a defining avian ratio. The alpha cells that produce glucagon outnumber the beta cells that produce insulin by exactly 2 to 1. That structural dominance proves a vital point. In birds, it is glucagon, not insulin, that serves as the primary dictator of metabolism and glycolysis. The dampened insulin signaling inside the cell is a necessary evolutionary counterweight to a pancreas that is physically built to flood the body with glucagon. So, what is the ultimate physiological outcome of a glucagon-dominant, insulin-refractory architecture? By heavily prioritizing lipolysis, the avian system guarantees the massive, rapid mobilization of energy stores from fat at a moment's notice. That rapid energy burn is exactly what is needed to maintain their unusually high native body temperature, which bypasses the standard mammalian 37 degrees and locks in at 42 degrees Celsius. That continuous availability of mobilized energy fuels the sustained extreme metabolic demands that are absolutely required for avian flight. And when they aren't in the air, this precise metabolic configuration allows birds to survive long catabolic states and periods of starvation without facing systemic collapse. This specific biochemical rewiring, which would be lethal in any other animal, is the exact energy management system that allows birds to rule the skies.